Hello, everyone. If you could all take your seat, we're going to get this panel rolling. I hope you all enjoyed the afternoon break of coffee and tea. I'd like to introduce our panel moderator this afternoon for Northern Mine Remediation and Restoration panel, Dr. Guillaume Nielsen. He's an industrial research chair in Northern Mine Remediation at the Yukon University.
Hello. All right. Thank you very much for attending this panel/session. I will explain that this is a panel/presentation. I understand very well that myself and the panel here, the panelists, are sitting just in front of you, between you and the end of Geoscience, and hopefully the many drinks that will follow. So thank you very much for attending this panel and giving it a last little push. And attending this panel were 6 panelists, including myself, professionals, students, researchers, people from the consulting world. We'll talk about mine remediation, mine restoration, and generally speaking about the environment. Before starting, I would like to acknowledge we are on Kwalindon First Nation and Tatquatchan Council traditional territory, especially today and in the last couple of days where we were presenting from that beautiful, beautiful building. I'll take a couple of minutes to introduce myself and also why this panel about environment, remediation, and restoration. So my name is Guillaume Nielsen. I'm the NSERC Industrial Research Chair in Northern Mine Remediation, and as I often say, that's a way too long title, but that comes with the job. I have to stick to it. And that means that I'm a researcher that partners with the mining industry. In my case, in Northern Mine Remediation, all the active mines in the Yukon, meaning the mines that are actively mining, shoveling the dirt, or that are being caught into the environmental process. You can see here that we have 6 mines. I still say 7 because Minto was part of it, not anymore, obviously, for the reasons you probably know. That still breaks my heart, so I still, at least I removed the logo, but I still have them over there. So 6 mines that constitute the Yukon Mining Research Consortium. So how do I work with this consortium? We meet 4 times per year minimum, and way more often with specific teams. And my chair is, they bring to me and to my research team environmental challenges they're facing that are specific to the North. And we must provide answers and scientific evidences to these guys to help them facing the challenges they're facing. 3 main research trends. The first one is water treatment by passive treatments. You probably know that a lot of the treatment, a lot of the water, mine-impacted water, is treated by what's called active treatments that rely on chemicals, labor, and big buildings. The passive treatments rely more on what Mother Earth can bring. So we're talking about bacteria and plants, phytoremediation and bioremediation. Mine waste management, especially the solid waste management, tailings and waste rocks. And last but not least, mine revegetation. So these are the 3 main research trends we're working on, and today we're going to present a couple of projects that are under these research trends. Community engagement is very important in my work, and I'm a YukonU employee. That means that I'm a neutral, independent researcher. We don't generate any IP, and all the knowledge and the research that we are generating must be publicly available and can be found on the Northern Mine Remediation website, on the YukonU website. So back to why this panel. So I'm originally from France. It's my 11th year in the Yukon, a bit more than 15 in Canada now. And since I'm part of this business, this professional mining world, you know, I realized that restoration, remediation, the environment, generally speaking, is not very well represented in mining conferences in general. And that's the case for big mining conferences as well, international Canadian ones. I'm not going to throw any names out there and throw them under the bus, but it's a fact. It's a lot about exploration, geological presentations, new discovery, business development, the return on investment, you know, And don't get me wrong, we need those critical minerals, and I'm quoting Minister Stryker that said exactly that in an amazing panel yesterday about energy transition. We need those minerals, we need mining, but we need also to talk about environment more and more. So I'm honored today to contribute to filling the gap a little bit. I'd like to thank Geoscience Organization Committee to giving us the time to discuss these important topics. And there is another quote from yesterday. I think it was in a panel about teaching and education, where someone said, "I hate to be honest, but I'll be honest." So I'll do the same. And I feel like the mining industry comes with a difficult legacy. We had a very good presentation on Sphero yesterday which is a very good example of what's a bad example from the past. And I attended many talks, probably as you. And yesterday I was listening to amazing panelists about economic reconciliation. And they said that the mining industry start to better understand what is the real meaning about return on investment, especially in the long term, when mining is such a volatile and fast industry. I feel like there is a shift. This is why we are more and more interested in engaging and involving First Nations into mining. This is why we are more and more interested in engaging with women in mining. I don't know if you attended the talk about Jenny this morning from Newmont, who was absolutely amazing about women in mining. This is why we are more and more interested about the impact, the environmental impact of mining. So I feel there is a shift. And this is why we're having that panel about remediation and restoration. And sometimes we're even past that. And what we are talking about, this is also the case today, is how to do it better, develop best environmental practices. So just to clarify the situation, I made it very confusing, my fault. This is not a panel, or this is a panel between a panel and technical talks. I hope you don't feel you're not tricked into technical talks. But I invited a couple of panelists slash presenters that will present about projects in a very technical way. Sometimes it's going to be too technical for some of you, or not enough, depending on your background. I think it's okay, it's not important. What I would like is to trigger discussions, interest about environment. How can we work better together from different sectors? We have people I haven't worked with in the past and I know about. We have students, we have researchers from the private sector, from the consulting companies. So how can we develop together those best environmental practices, and how can we support our beautiful territory? I'll act as a facilitator, and I'll be the first one to go talking about one of the pilot scale experiments we conducted in the last couple of years, called the pilot scale bioreactors at Eagle Gold Mine. We especially wanted to assess how freeze and thaw cycles impact the bacteria and their efficiency to remove heavy metals. So as a basic reminder, acid mine drainage is one of the biggest environmental challenge the mining industry is facing. It occurs when sulfidic minerals are exposed suddenly to both oxygen in air and water, and it generates three things: acidity, sulfate, and heavy metals. The passive treatment system available to handle this kind of contamination is called sulfate-reducing passive bioreactors. And how does it work? First of all, it consumes sulfate. That looks like a good news because sulfate is one of the byproducts of acid rock drainage, acid mine drainage. On the other hand, it produces sulfide, and then that sulfide precipitates as a metal sulfide. And if I say that again, but with a little bit of chemistry, the water, mine-impacted water with sulfate generates sulfide, and that sulfide precipitates with the heavy metal. It settles down, it's removed from the water. But there is a blank here because we need a secret weapon in those bioreactors. We need sulfate-reducing bacteria. We need the bugs to do it. And we need carbon. I'm going to talk about that a bit. So, quick introduction. This study was not thrown out there at Eagle Gold site. It comes from two major studies conducted before. One in a lab-scale study conducted at the Yukon College at the time in 2014, and a pilot-scale study, that was actually my PhD work, conducted at Keno Hill in 2014. I could spend hours talking about these two studies. I won't. That's not the case today. That's not the goal for today. What the main conclusions out of it is we found that those native from the Yukon bacteria can successfully remove bivalent metals, heavy metals, from mine-impacted water. And we found also, following a genomic characterization, that that microbial community can survive over the winter, even if it's very cold, but the temperature impacts their efficiency to remove heavy metals. There's a couple of reports available online at the Northern Mine Remediation website, and there's a peer-reviewed publication about that work as well. Back to what we want to discuss about today. So, following these two studies, and with my new role as the chair, the industry came up with a question, very important question, very applied question from the real world. They said, well, it was Victor Gold, and you can see a picture here of a map. You can see that down there is the Platinum Gulch waste rock storage facility. And they said, we have a drain collecting the water from that waste rock pile, and that drain freezes in the winter. There is no water being produced. It freezes. What happens if we decide to go with your technology and treat the water? What happens to your bioreactors if they freeze? Because obviously it needs to be a passive treatment system. Do we have to heat the bioreactor? Not very cost competitive. Do we have to feed them with sulfate and carbon to keep the bacteria alive? That would make it challenging. So what happens if we let them freeze? How does the freeze and thaw cycle, how do them impact the microbial population and subsequently their efficiency to remove contamination of concern, in that case, antimony and selenium. So I looked at the literature and it was not too much, so we designed that study. It looks like that. We have 2 duplicate bioreactors into a shed. Why into a shed? Because they were prevented from freezing. The shed was maintained all year long at 5 degrees Celsius. And 2 bioreactors, the same one, outside of the shed. So pilot scale study, we're talking about 55 gallons bioreactors. And those one outside were following freeze and thaw cycles. That's a study that we conducted for 3 years. And some of the graph and charts you'll see are for 3 years. Some of them are for 1 year because we only had budget and time to do the genomic characterization over 1 year. So I will navigate you through this. So I referred in the next in the presentation as bioreactor 1, duplicates inside, 2, duplicates outside of the of the shed. So how does it look like, bioreactor composition? It is very simple because it's a passive technology. It has to be simple. So we sample bacteria. I'll have a slide about the bacteria. We sample bacteria on site. They have to be native from the mine site. Why? Because those bacteria are cold adapted already. They are the one you want into your bioreactors. They are the one that are the most relevant to handle the contamination that you have on site. Wood chips because it's a carbon source and it's also a good growth surface for the bacteria. And that's pretty much it. Then you run the water into it. What you see here is what we call tea bags. It's a bacterial sample that we use to do the genomic characterization in the future. So I have a slide about sulfate-reducing bacteria because obviously they are absolutely key in the process. And those bugs are very cool. They use sulfate as final electron acceptors, and they reduce sulfate into sulfide, as I said. They're ubiquitous, which means you can find them absolutely everywhere on Earth. One and one only conditions, anoxic. They don't like oxygen. So you saw a picture of me shoveling creek sediment. This is where, for example, you can find those bacteria. Wetlands, where it smells very bad. Sulfide, actually, rotten eggs, are very good candidates to find those native bacteria. They have optimal conditions. pH neutral, they need carbon, they are usually mesophilic, they like room temperature, but these are optimal conditions. We can work around that. We can mitigate those conditions, especially, for example, to make them work in the cold of the Yukon. Food is absolutely key in the process to mitigate the cold. I won't talk too much about food and what carbon means to the bacteria because Taylor, who will follow me on a presentation about how she used bacteria, she has a couple of slides about that. And now, let's dive into the results. There's 2 ways to understand what's happening into the bioreactors. You study the chemistry of the water, in and out, and then use the ultimate tool, the genomic. So the chemistry, first of all, temperature and conductivity. As you can see on the first graph, the temperature over 1 year it goes down to -50 outside. So obviously in the winter, those bioreactors outside, they did freeze. And the temperature in the winter is set at 5 degrees Celsius. This is the triangle in the bioreactor 1, the one inside the shed. The conductivity, I just wanted to show it because it shows something very important, which is the fluctuation of the chemistry in the water depending the dilution concentration effect following winter versus summer. Basically, this is how much salt, conductivity is how much salt you have in the water, and as you can see, this is pretty concentrated in the winter, and it's pretty high in the summer. So you have a dilution concentration effect, and this is why those pilot scale studies are important for the mining industry. This is why those pilot scale studies implemented on site are important for the regulators, because we are working with real mine impacted water, real chemistry, real hydraulic retention time, real flow rate, real everything. So this is one step ahead for the demonstration, for the feasibility of such technologies. When I look at the chemistry again, one of the very good ways to understand if the sulfate-reducing bacteria are working is to have a look at the sulfate concentration. Does something eat it? If there is any chemist in the room, they don't eat it. They will know. So do they eat it? What you can see here is, on the figure 3, is the concentration, and then the average sulfate reduction, so the removal of sulfate. Obviously, you can see that on bioreactor 1, the top graph, I should show you over there, not on the screen. On the top graph over here, top right corner, you see the removal of sulfate in the bioreactor once, inside the shed. The highest removal percentage occurred in August, and we removed 97% of the sulfate with the sulfate-reducing bacteria. So we have bacterial activity. The chemistry tell us that. There's a lot of things we have to check, like calcium, to make sure it's not gypsum precipitation, but that tells us that we have a microbial community. And then look at the one below. There are missing spots, missing blocks in the bioreactor 2s. Why? Because over 3 years, there is freeze and thaw cycles. When it's frozen, we cannot sample it. Nothing happens. But when they thaw those bioreactors, without doing anything, when they thaw, look at that, they catch up. Within 2 weeks, the removal rates of sulfate are very similar to the one in bioreactor 1. The bacteria first of all, appear to survive, and 2, go back to work, and 3, go back to work quite fast. So that's the first pretty cool preliminary results. Subsequently, the game is obviously to remove the contamination, the heavy metals. Antimony concentrations, I'll just give you the numbers for sake of time. We removed up to 96% of the antimony with the sulfate-reducing bacteria and sulfate-reducing bacteria only. We removed the selenium up to 94% with the same process. So that was the chemistry. Now let's dig into the genomic, which is, as I said, the ultimate tool. The genomic, we had 3 questions. Question number 1 is, are the bioreactor 1 inside and 2 outside average bacterial population identical during the summer, the first summer of the study? Meaning, did we use the same inoculum? That's the first question. 2, are the bioreactor 2 and 2Bs, those duplicates, outside identical before winter and after winter? That question means, did the freezing cycle impact the bacterial population? And finally, are the bioreactor 1 and 2, inside and outside, composition identical during summer 2023, meaning did they evaluate differently after the freeze and thaw cycles? I have a couple of pictures to illustrate another important component of this work, which is how challenging it is to conduct this study. I mean, we're northern researchers, we're based in the Yukon, but taking those samples, it's still 6-7 hours driving up and then same down. So you have to, in order to tell those bacteria, until you do the genomic characterization and DNA extraction, to say, you stay as you are right now, because I need you to stay to understand who you are right now. You need to freeze them into liquid nitrogen, minus 200 degrees Celsius. You have to drive as fast as the wind, following speed limitation, to the lab. And then into the lab, you have to freeze them again until you have them into a minus 86 freezer. What you see here is a picture of my boy helping me doing the last. He's absolutely not wearing the appropriate PPE to do that, so May's cute, so I kept the picture. Then comes the time to do the DNA extraction. So we usually partner with UBC to do this. We developed this year the capacity to do it into our lab at UConn University. And then we generate these fancy charts. So these charts are beautiful. They tell you exactly what bacteria you have and the relative abundance in your samples over time, obviously. 2 reasons I'm not going to go over it today, even if it's like very beautiful. As I said, it's like art to me. We don't have time. And 2, we have a peer-reviewed publication that is submitted, so I don't want to steal my own thunder. Though what I can do is share conclusions about these 1-year genomic characterization project or 3 years total length project. One, the bioreactors, which is the one inside the shed and maintained at temperature, 5 degrees-ish, removed successfully selenium and antimony consistently over the year. The bioreactor 2, which are the one outside of the shed, presented very similar behavior before the freezing event. Obviously, during the spring and thaw period, the percentage of removal was slightly smaller. It took a bit of time for those bacteria to readapt. However, at the end of the thaw period, and during summer and fall, the removal percentage reached similar value. They caught up. Surprisingly, we have some variability, that's the genomic part, about the relative abundance in the microbial population. However, that doesn't impact the bioreactor's removal overall. This study shows that the freeze and thaw cycles affects the relative abundance, as I said, but the removal capacity of the bioreactors is not impacted substantially. The bacterial consortium growing, which are the ones naturally present on-site, can work in subarctic regions. Thank you very much. We have industrial report available online and the peer-reviewed paper to come. Thanks. So, as I said, it's a mix between a panel and a presentation. So I would encourage you, obviously, to ask questions. I would like the panelists, who some of them don't know each other, to ask questions as well. So I'm pointing at you. Sorry about that. But if you have questions, please. We'll be also presenting the posters after this at the closing ceremony. But if you have any questions, I'll be happy to take them, and also I'll ask questions to the rest of the panelists. Very clear? Thank you.